Welcome to the Qualitas podcast. I'm Jan Kugler, your host, and my guest today is Chris Maughan. Chris is an expert on sterilization and validation, and we're going to talk to him today about autoclaves. It seems very simple, but issues with this equipment can cost you a lot of time and money. It can set back your production weeks, even months. So it's crucial to avoid it. So we're going to talk with Chris about how to choose the right equipment for you, how to plan the validation phase correctly, and how to think about the correct user requirement specification and test everything so you don't have any issues down the road, because it's much more difficult to fix it afterwards. So let's get to it. Chris, good morning. Welcome to the Qualitox podcast. Great to have you on the show.
Good morning, Jan. Good morning.
So today we want to talk about autoclaves, their purpose in the pharmaceutical industry, and how to choose them, how to do the right and correct validation and qualification of the equipment, how to choose the vendors. So it's quite an interesting topic around it. So what is your expertise in the field?
Okay, so I've been working with Autoclaves, specifically within validation, since 2003, so quite a while. I set up our company Thermal Compliance in 2007 with a focus on steam sterilization and particularly autoclaves. So we get involved throughout the project, right from the initial phase of URSs and then all the way through to qualification and requalification. So I've got quite a good background in the validation of the process and also from an engineering perspective as well.
What are the uses for autoclave? So let's go maybe a bit into general if we have more newcomers to the industry. So in the pharma industry, where do we use autoclaves?
Okay, so we'll use autoclaves in the micro lab, for example. So that might be to sterilize any media that we're using within the micro lab. We'll use it within production to sterilize equipment that's used in creating the product. So it might be vessels, it could be filters, pipework, it could be kind of forceps or anything that's used in the process during the production, and also the terminal sterilization of fluids products as well. So they'll go through the autoclave, and what we're looking to do is to kill any microorganisms that might be present within the load that goes into the autoclave.
So what are the major types of autoclaves that we see on the market right now?
Well, there's really two main types. So we've got a fluids load autoclave, and that's used for terminal sterilization of things like syringes and bottles and final products. So they're sealed containers with an aqueous solution inside, so a liquid water-based solution. And essentially, it's generating its own steam inside that container. And then the other main type of an autoclave is a porous load sterilizer. And essentially, that's items that are being placed within the autoclave and we're sterilizing the surface of them. So they might be instruments that are used during production that would come into contact with the product. So there's two main types of fluids and porous load autoclaves, and they have slightly different ways of setting them up to make sure that it's suitable to your process.
Okay, and when you choose one, so there are two main types, but probably there are a lot of vendors. So what sets the vendors apart?
Yeah, sure. Well, it really depends on what your process is. So I think, and this is quite a difficult thing to do with any facility, but you need to understand how you're going to be producing your product. What kind of equipment is the autoclave going to be used to process? So if we can understand that as early as possible, we can start to formulate what kind of functionality we need from that autoclave. So if it's a porous load autoclave, for example, what loads is it going to be processing? Are they going to be difficult to sterilize? Are they going to be difficult to dry? Do we need any added functionality? So if it's a porous load autoclave and we think it might be wet, then we can start looking at having hot air drying to enable drying conditions. We can look at vacuum pumps. And for fluids loads, we might be looking at having fans installed to give us good circulation within the autoclave. So we're trying to understand what it's going to be used for. Often autoclaves are called multi-cycle autoclaves, and they're used for porous and fluids loads. So I think before you start looking at manufacturers, it's important to understand exactly what it's going to be used for and the complications that we might see going down the line. So if we anticipate issues during the validation stage, we can anticipate that before we go through with that purchase. So before the URS is written, we write that robustly because we know what we're going to be using it for.
Would you start looking into it already before you go to the user requirement specification, the URS, or would it be already a part of it?
I think at that stage, you really want to get somebody into the process who understands what's happening. So it's very easy to order an autoclave. This company manufactures autoclaves. That's the autoclave we're going to buy. That's within our budget. But actually, I think to prevent any issues further down the line, it's much better to understand how are we going to be using it? What are the likely pitfalls during the qualification stage? And how is the process going to work? How are we going to load it? And when you can understand all of that information, you can then start filling out your URS and saying, we're going to need this additional functionality. Because typically autoclaves come as an autoclave and they'll come with functionality to sterilize the loads. But you can reduce the length of the cycle on the autoclave, you can improve the air removal, and you can improve the drying at the end of the cycle, all by specifying the right equipment at the right time. But it's not always necessary, so it doesn't usually come as standard.
Right. So you mean there are autoclaves and there are vendors that are ready to do custom made machines and do software and hardware customization?
Absolutely, yeah. So most of the big manufacturers will be able to specify your autoclave for your application, and that would be whether you're meeting sort of a standard like EN 285, which a European standard which focuses on air removal, and some of those aspects would include air detectors, which are used predominantly really in the UK and Ireland, but you would need to specify that again in your URS. So that's kind of a basic example of something that you would need from a local perspective. But sort of on the broader scale, there will be issues potentially with wet loads, with air removal, that can be rectified if you do that before you purchase the machine. And again, that's working with your suppliers to ensure that you get that.
Right. So it means that you probably should have engineers and experts that really understand
Absolutely,
how the physics works.
Yeah, I'm a big advocate of bringing in validation as early as possible and engineering,
Right.
because these are the guys that work on the machines. They probably already work at them in your facility and they understand how they work. They understand some of the issues that we're likely to face further down the line. And it's a strange issue, but if we can iron those issues out initially, an extra couple of weeks' work at the URS stage will result in far less issues in the future for ongoing production. And also it'll allow you to use that autoclave continuously with less downtime, because you won't be seeing some of the issues because you spec'd it correctly from day one, really.
Exactly. So what are the biggest issues or the mistakes that companies do when they purchase the autoclaves? You mentioned once that they look at the budget that they have and then they buy it according to the budget instead of the direct use requirements or their processes. Is there anything else that people should be aware of when they're choosing them?
Well, yeah, I mean, I guess the budget is more. It's an autoclave, so we'll buy that one. We have X amount of money to spend. That's how the purchase decisions are often made. Obviously, we need to meet the regulations, and they're usually included in the URS. So the things like the materials of construction, all the certificates, the welding logs, all of that information is recorded, and most manufacturers would issue that standard. It comes when we're working with complex loads. So if we've got some really complex loads that we're going to be placing through the autoclave, if we the type of wrapping that we're going to be used to wrap the load items that go into the autoclave, all of these can have an impact on the validation and on the continued use. So if we have people who understand that, then we can sort of set the autoclave up to function correctly from day one, really.
How do you write a good URS? What should it contain?
Well, I think it's about understanding, like, for example, you mentioned fluids. So product formats, if you've got a very large fluid format, so let's say it's a litre of product that's being sterilised, the question then would be, how can we make sure that that cycle doesn't last for sort of 8 or 9 hours? And if we can reduce that cycle time, but still maintain that sterilisation efficiency, so we can have the same level of kill, but a shorter time frame on the cycle, then it's perhaps not a quality issue, but we can increase productivity of that machine. So once you've purchased that autoclave, it's very difficult to retrofit some of the cooling cycles, for example, that might help cool that load. Because a lot of the cycle time is taken up with not heating it up, but bringing it back to a safe temperature so you can open the autoclave. So we would want to look at that stage at what kind of additional capability can we add to the machine to allow those loads to cool faster. So that might involve spending extra money at the startup phase, but that could save you weeks over a year in production timelines that you would lose for having a very long cycle. So it's a mixture of anticipating exactly what it's going to be used for and how we can help reduce sort of timelines, how we can help reduce the potential for failures by improving various aspects. So for fluids loads, that's often the cooling phase. So the heat up phase is normally quite good. But for porous loads, that could be the air removal phase. So if we have a complex load with lots of thin bore tubing, or we have a tool can that might fit onto an isolator, for example, they need to remove air from that load that's kept within inside a tool can that might go through a 0.2 micron filter. So we need to start thinking then about the vacuum pump that we're using. We need to think about how we can control the rate at which vacuum pulses happen on the autoclave. We need to think about how we're going to remove any excess moisture, about how we can keep heat within the load during the cooling phase to flash off any potential residual moisture at the end of the cycle. So there's quite a few complex issues that can be addressed with a good URS at the very front. So that would be looking at vacuum pulses, at any additional drying cycle that we can add, any overpressure. So that might be putting additional services onto the autoclave, like hot air or compressed air. It might be having cooling water to the autoclave, just to enable us to cool loads, to remove air, and to dry loads. So it's something that's very difficult to retrofit. So we can do that at the URS stage because we can anticipate it, I think.
Right. So it sounds like a lot of playing around with the functions and with the engineering of it, a lot of simulations. So how do you perform it basically? With what parameters do you need to look at? So are we talking about engineering that taking into account the volume, the amount of the equipment or the materials that you put inside, and the size of them and the material they're made of, and that all comes to the circulation and the parameters of the device, and then you see how you can adjust or change it, right?
Yeah, exactly. I mean, the load items themselves will affect the issues that we have, and also how they're wrapped. So, for example, if we're transferring heavy stopper bowls on an isolator from the autoclave to the isolator itself, and how we transfer that, going from the grade A of the autoclave through then to the grade A of the isolator, that transfer is really important. So we often use Tyvek bags, for example, and Tyvek bags are great for letting steam into the autoclave, through the bag to the item that we're sterilizing, but they're quite bad at removing water from that bag. So we end up with small pools of condensate. So that's quite a common issue that we'll see on those kind of loads. And then we've got to think about how we can remove that. So we need as many tools in our toolbox within the autoclave's fabrication to do that as possible. So if the URS hasn't thought about that at the very early stages, we can end up with extremely long cycle times and perhaps needing to replace the autoclave because we haven't considered how we're going to use it, what loads we have, and what potential issues we might face. So it's the same with the fluid cycle, for example. We're looking at it from a different angle, but we've got issues with that cycle that we can improve quality and improve the sterilization and improve timelines. So all of those issues can be resolved with a little bit of consideration at the URS stage, a little bit of engineering input, a little bit of validation input can all help.
So after people and the engineers did their job, they written the URS, they discussed everything with the manufacturer, they got all the information back, it looks good. What are the next stages?
Well, once the autoclave is constructed, you might do an FAT. So you go to the factory and run various tests to make sure the functionality is correct. You take a look at the software, at air removal. So you might do some basic tests on the machine within the factory. And assuming everything's okay and any issues are identified at that stage, you then move on to a site acceptance test where you'll install it and then check everything's functional, and you'll move on to IQ and installation qualification. So at that stage, we'll look at everything to do with the installation, to do with the materials of construction. Essentially, that's everything before we turn it on, I guess. So we want to see things like weld logs and the wiring, has that been done correctly? So we'll look over the whole machine. We'll walk through the process drawings and make sure that they're all correct as well. And once that's completed, we can then move on to the operational qualification. And during that phase, we'll look at things like the alarms. We'll look at the different aspects of how the machine functions, the cycles that are already installed. We'll test them. And we might do some additional work to test air removal. So are the cycles robust enough for the loads that we anticipate using? So we use tests that are detailed in EN 285 for that, because they challenge the autoclave cycles rigorously. So assuming we can meet that criteria, we're at a good position and a good baseline to develop the autoclave cycles for the production loads. So once we've closed out the OQ and we've done all of that testing, we then move on to cycle development. Now, this is something that's not always performed, but it's recommended, particularly for sort of complex loads, for fluids loads. We need to understand a few things, really. First of all, are we sterilizing all of the locations within a load? So we'll look at actual loads that are going to be used for production. We want to make sure that things like long lengths of tubing, filters, anything that we might expect to cause us an issue, is being sterilized effectively. So we're getting the right amount of time and temperature. And to do that, we can then start tweaking the cycle to make sure that it's set up for those specific loads, that we're getting the best results possible from that. And once we're confident that we've got all of that data, we can also use the cycle development phase to look for any cold spots. So if we've got slow to heat locations or cold locations within the chamber, that would indicate that we're not getting proper sterilization conditions. So we can then identify any of those locations, and we would use those going forward for the next step, which is the performance qualification. And that's where we would map in triplicate the load formats. So there might be multiple load formats. Each one would be mapped 3 times, and that would then qualify that load for use. And after that, it would be an annual requalification of each load to ensure that it's still meeting the requirements.
So how do you do the mapping? What are the techniques for that?
For the mapping, we want to use temperature sensors, and we can either use sort of wired thermocouple type sensors, or we can use data loggers, wireless data loggers that are becoming increasingly popular. And we place them throughout the autoclave loads in areas that we would assume to be worst case. So we would place them within like long lengths of tubing, within filters, within vessels, and we would try and map every single area within the chamber. So by that, I mean every single load item. And with each load item, we would look for that worst case location. And we would usually base that on air removal. So if it's hard to remove air from the load, we would place it in the areas that we deem to be hard to remove air from. And in addition to using these the thermocouple sensors, we would also use biological indicators. So that would have an organism on like Bacillus staphylococcus. We would take them from the micro lab, place them in those hard to sterilize locations, put the load in the autoclave, run a cycle through, take them out again, and then try and grow them up. So we put them in an incubator and try and grow them up over time to see if we can. If we can, obviously that's a non-sterile load. We haven't been able to demonstrate sterility.
Do you do both of them, also sensors and the microbiological tests, or they're interchangeable?
Yeah, normally for cycle development, we would focus on temperature only. So we're looking for those difficult to sterilize locations. But when you move into performance qualification and requalification, they go side by side. So you'd place a biological indicator next to a temperature sensor. And I guess that way, if we did see growth in a biological indicator, we can then look at the temperature data and say, look, did we have good air removal? Did we have time at temperature? Is there another issue here? Because steam sterilization is about hitting the sweet spot with the quality of the steam. It's very important that the steam's not too dry. The organisms are killed by moist heat. So what we need to make sure is the moisture that's within the steam, that's what breaks down the cell wall of microorganisms and bacteria. So if it's dry heat, it takes a lot longer to kill. So if you can think about, if you've had any experience of working with deep hydrogenation tunnels, they operate at a higher temperature, generally for longer periods of time than the steam sterilizer. So we might be at 300 degrees rather than 121 degrees. And it's really because moist heat kills a lot faster than dry heat. So high temperature in an autoclave is not always a good thing. So we could have a high temperature and no BI kill potentially if it was dry heat. So we're looking for both. It's a combination. I mean, historically, there's been a bias towards biological indicators in the US and within Europe. We've looked more at kind of engineering and at temperature and thermal data. But I think the two of them together, it sort of supports biological kill, which demonstrates that we've got lethality within the autoclave, and thermal data, which gives us that engineering information as well.
Right. And are there also some kinds of worst case scenario testings?
Yeah, there's routine testing that we perform. So we'll perform things like Bowie dip tests. So I've got one of those here. It's these go in the autoclave and they have a chemical indicator inside. So we run these on a daily basis. And that chemical indicator changes from, in this case, from a sort of yellow color to a blue color. And that tells us that we've got good air removal. So we've got good steam penetration into that pack. We're also concerned with any leaks into the chamber. So if any air is leaking into the chamber, that can affect the sterilization. So we run a leak rate test. That's normally performed once a week or on a daily basis. And that'll give us any information to, is there an issue with the integrity of the chamber that could affect the sterilization process? We also have in the UK and Ireland particularly, we have something called an air detector, which can detect any air within the chamber. They're not commonly used outside the UK and Ireland, though, so it's more a kind of localized thing. But there is various methods. Also, during production, you can use chemical indicators or biological indicators that you can place within the autoclave, and that would then give you an indication that you've had sterilization conditions. So there's various tools that we can use, but really the validation stage, that qualification stage, using the biological indicators and the temperature sensors, that's going to give us the most robust information and kind of qualify that load.
So what would you suggest that companies never skip or always do from your experience? If you see, okay, they haven't performed that or they didn't put enough work into this stage, then they have those and those problems which cause them a lot of time during the production, a lot of deviations. Are there such steps which sometimes are overseen and cause problems?
There's a few issues that sort of regular pitfalls that people fall into and regular issues. And one would be wet loads. They're easily overlooked. There used to be a phrase that was sensibly dry for an autoclave load, which is kind of objective because people would say, well, it's only a little bit of water, that's relatively sensible. I think the meaning from the original standard was it's detectable by the senses. So if you can see it, if you can touch it, if you can feel it, then the load is wet. And with wet loads, what we're concerned about is that can easily get recontaminated with microorganisms. So something like that is an issue, and it's something to be aware of. And it could also be an issue with the quality of the steam that we're using in the autoclave as well. That could affect that wet load. So I think for me, it's important that the weekly tests are performed, that we're running leak rates, we're doing preventative maintenance on the machine, and that we've got regular validation that's looking into those load items and looking into the complex areas and making sure that we haven't got any issues with air removal and that everywhere's being effectively sterilised. But on a basic level, I think people should be using biological indicators and temperature sensors and placing them correctly in the worst case load areas.
Right.
As a minimum, really.
Right. So, Chris, so to summarize a bit the consequences, right? So we talked there, it seems that qualification of the autoclaves and even the choosing the right ones is quite big work, and it's quite complicated and should be taken lightly. So what are the biggest issues if you don't follow all this and do it incorrectly? So you mentioned if you don't design it correctly, you can lose days, even weeks of production time, which really costs you a lot of money. So this is one. Then you can get much more deviations on your head, which means you need to do much more reworks and documentation. And again, your production is has a big step back, and you again lose money. Is there anything else?
Yeah, I guess when we're looking at that, if we do have a production failure and it's a regular failure, it's an awful lot of time. Your machine can be out for a week without being able to use it at all, and that could happen fairly regularly. And it's all about first of all, with the URS, ensuring that doesn't happen, and that we've got that kind of we're aware at the URS stage that we can put something in there to prevent it from happening, or if it does happen, to help resolve it. But also it's about understanding any failures that you do get and understanding the implications of any changes that you make. And that's where we see a common pitfall is there might be an issue with the equilibration time, which is I haven't really gone into that, but it's part of the data that we're looking for to make sure that all of the temperatures within the chamber come up to temperature within the required time. So that's either 15 seconds or 30 seconds, depending on the size of the autoclave. But if that fails, there are various ways that you can fix it. But there is the right way to do it, and then there are other ways that can resolve the issue, but can lead to potential future issues. So it's about understanding any changes that you make to an autoclave cycle and the potential impact that might have on the future. And it's very difficult to get that message across for people to understand that you really need to understand exactly what you're changing on an autoclave to ensure that it continues to function correctly. And you're not saving up issues for later on down the line, really.
Right. So it's a really big process. A lot of background knowledge should be there, and there should be SMEs on the spot, because it's not just a production issue, a QA issue. You really need to understand how this simple equipment works. You know, it should be, but it's not that simple.
But it is, like you say, it's a really simple piece of equipment. All it is a box with steam in it. But it causes a lot of problems. And anybody who runs autoclaves will know there are always issues. And it's about minimizing those issues and preventing any continuous downtime and ensuring the quality as well throughout. And I guess detecting any failures. You know, that's ultimately, nobody wants any non-sterile units going out of the door. You know, we want to make sure that they're all sterile and we have confidence in it. So we need to make sure that we're detecting any failures as they might arise, and we're preventing any issues that might cause equipment downtime. So we're optimizing the process, really. That's particularly with the URS, I think that's really important, that we optimize that process, get it set up so it can perform and function correctly, as well as possible, as fast as possible, and to the right standards as well.
Right. And to help people with that, you have also created several of free workshops, and that should help people with those issues, right?
Yeah, absolutely. So we've got a few on things like we mentioned wet loads earlier on. There's some of those that people can watch. So it's the workshops, webinars. It's called Validation Workshop. It's all on LinkedIn, and people can access it on there. So if you just search for Validation Workshop on there, you can access all of the back catalogue. And hopefully, if you do have any issues like that on site, you can just sort of watch a webinar, and it should point you in the right direction. But yeah, I think it's useful to have that information out there. So I'm hoping that people enjoy it and find it useful. But yeah, it's been going for nearly 10 months now, something like that. And it's good fun to do. And I think people enjoy visiting. So,
so it's a live one, and you also have recordings?
That's right, yeah. So they're live. We do them once a month. They're on various topics, and people can vote for whichever topic they prefer. And they're once a month, usually on the first Thursday of the month. And you can rewatch them on our website, which is thermalcompliance.com, and look at the back catalogue on there as well.
Okay, perfect. So I really recommend people to come and visit those workshops, especially as they're complementary. And I think you can learn much from that and save a lot of pain down the line, right?
Yeah, absolutely. Yeah, hopefully.
Chris, thank you again for this talk and for bringing this autoclave world and showing how complex it can be if you don't know what you need to do.
Yeah, no problem. No problem. I hope people found it useful.
Thank you very much.
Thank you.